Good evening. My name is Rani Avisar. I'm the dean of the school for another few months. And this is my 16th and last year as hosting this series. And it was created. This is the 30th year of the series. It was created by late Professor Ginsberg, and since then have been consistently every year brought here 4, 5, 6 sometimes lecturers. Originally mostly from outside the university, but over the years we have improved that, I would say, by including some of our own lecturers, as is going to be the case this evening. So welcome everybody here in the auditorium and online. And this evening we are going to hear Dr. Catherine Macdonald. Her lecture is on biology, ecology, and conservation of sharks and rays in South Florida. Obviously a topic of great interest, okay, from very young kids to senior people like me. And before we start, though, I'd like to acknowledge the sponsorship of different organizations and individuals for this series, certainly over the years and more recently. And the presenting sponsor this evening is the Norwegian Cruise Line Holding and the Shepard Broad Foundation. We have also the contributions for the series of William Galway III, Cheryl Gold, who is here in the audience and is not only contributing to support the series, but is also contributing tremendously by doing the research to identify speakers, especially outside of the school, and has done over the years a fabulous job of identifying speakers that are most of the time, part of the actual environment going on. So this year, obviously, we will we'll continue having lectures on different topics, including, in fact, fire, natural fires, and which is obviously very relevant given what's happening currently in California. In addition to Cheryl, we have also the KB Life Enhancement Forum contributing to our series, the Key Biscayne Community Foundation, the John McCaffin Family Foundation, Nicole and Myron Wang, and I just saw Myron also in the audience. Welcome and thanks again. And the Southern Glazers Wines and Spirit. Over the years, we have also established kind of a tradition to spotlight not only the speaker, but alumnus of the school, as well as graduate students, active graduate students. And tonight, I am delighted to introduce to you an alum, Aaron Stauber, who exemplifies the lifelong learning. Occasionally, we have, in fact, a few individuals that are coming back to school after a very successful career in a completely different area. And this is the case with Aaron, who graduated, in fact, with a JD from Georgetown University a few years ago, I would say. And after, I would call that a very successful career in commercial real estate, has decided that that was not enough, and he came back and studied at the school, got a master's degree, and we'll hear a little bit more about him in a few minutes. As a real estate company, he created the Rugby Realty Group, and he was directly responsible for the purchase and redevelopment of more than 10 million square foot, and with a market value of over $1 billion. He raised his family in the New York area and moved back. He was originally from South Florida and moved back to South Florida in 2015. He's a scuba diver and kiteboarder, and very interested in unsustainable development and climate change. And as I mentioned, in 2018, Aaron came back and got his master's from the school. He really completed an internship and created a septic tank pollution campaign for Miami Waterkeeper, where he now serves on the board. Aaron, please.
Thank you very much, Dean Alvisar, and thank you very much to everyone who's here. I've been given 5 minutes to get through 13 slides. That's 25 seconds per slide. And in 5 minutes, I'm going to explain that. Aaron Stauber's journey from kiteboarding to septic tanks, also known as Aaron in Wonderland. Okay, ready? Here we go. We already heard the background, real estate developer. So for 35 years, that's what I've been doing. Back out, I guess, since 2018. So roughly 6 years. With 29 years, I've been on the other side of the table of now me, which I used to call me the tree huggers, which I viewed as impediments to what I wanted to do. And I used to think I'm crazy. So in 2017, I moved back down here. In 2015, I got into kiteboarding with my brother Daniel. That's me on the right. That's Daniel on the left. He's my older brother. He's 68. So we would kiteboard here at Crandon Beach and also at Virginia Key. That's at Virginia Key. It's beautiful. And all of a sudden, we noticed about every once a week, they'd shut down the beach. And I'd ask somebody, why are they shutting down the beach? And they'd say, well, because it's polluted. It's unsafe to swim in the water because it has high levels of fecal coliform, which I had no idea what that was. I said, are you kidding me? This is the Atlantic Ocean, okay? I mean, it's not supposed to be polluted. So I was really curious about that. And every day when I came to Crandon, I'd pass by this building. And I figured they have the answer. I'll just go walking in there and I'll ask them, and then I'll solve it, because that's what I do in my business. So I walked in, and by the way, you can fact check this, so I'm not making this up. I walked into the dean's office, and I asked the secretary if there's somebody I could speak to ask them how come they're closing down the Atlantic Ocean. And they said, wait here for a few minutes. They invited me in. I don't remember if it was Dean Alvisar. I don't remember. I sat down in the chair, which was remarkable. They let me in. I was in my board shorts and my flip-flops because I was on the way to go kiting. And I asked the question. They said, and by the way, this is going to be a theme for, I think, Professor Macdonald's conversation tonight. The answer I got was, it's complicated. Oh, well. So they gave me a quick tour of the campus. I was really enthralled with the wave. They had just finished the wave room, which is remarkable. If you haven't seen it, it's in a building right over there, which is crazy. They can reproduce hurricane force conditions and see how it affects coral reefs. And it shows you basically the benefits of coral reefs. So if you question and doubt the benefits of coral reef, just step into that building over there and they'll reproduce it for you. So I finished the tour and I said, okay, I'm gonna go back, I'll register. I did, I got accepted remarkably. And this is the next thing. So that's my parking pass from 2018. Here I am. And then I set off on my Wonderland journey. And so I'm just going to take you for a very quick tour through that. So I had all the day classes. You know, I had done the law school thing, so that wasn't so exciting for me. But this stuff was amazing. So I did 2 separate trips. I did one with Professor Fernando Bretos, 10 days in the western part of the Yucatan Peninsula. most of us will never go to the western part of the Yucatan Peninsula. It's on the other side of Cancun. You probably don't know. I would never have known it exists. I landed in Merida, and I'm like, whoa, this is really cool. And then we went further west, and we went to the coastline, and we started going here. This is the Celestone Biosphere. And if you want to see what Biscayne Bay used to look like before Brickell Avenue was Brickell Avenue and Miami Beach was Miami Beach, you have to go here, go to this biosphere. That's an estuarian bay, exactly the way we were, but before we did what we did to it. So it's got all the mangroves, it's got all the seagrass. It's migratory landing spot for probably hundreds of thousands of beautiful flamingos. It's breathtaking. So that's, and it's, by the way, it's about, I don't know, a half an hour flight from Miami or something like that. It's crazy. And then what we did was, oh, that's a mangrove restoration area that you'll get to see. You won't see it here, but you'll see it there. And that's unfortunate. And then we visited 5 different marine academies in the Yucatan Peninsula. And we sat down and we met with the different graduate students there. And the one thing that blew my mind there was that the students that are there are not paying anything for school. If you want to go to school in Mexico, which I haven't checked, but it probably has a less GDP than the United States of America, you don't pay a thing. You just go to graduate school. And it's amazing. But I also learned another thing there, and then I learned it here, which is this laboratory that we went to, which farms, they learned how to farm octopus, which it turns out is a great sustainable fish. But I don't like octopus, but apparently it's good eating. What I learned there were 2 things. One was that because they have terrible pollution problem, very similar to what we have. They don't have a sewer system basically in the entire west part of the Yucatan. So they only use septic tanks. And I learned all about the karst system, that we have a very porous substrata. And but their problem is they don't have the money to do anything about it. They'd love to do something about it. In fact, it's first and foremost on their mind is environment, but they legitimately don't have the money. So they're more concerned that what they do have money for is putting food on the table, you know, providing education to their children. But that's the west part of rural Mexico, not, you know, Miami Beach, Florida, and Miami-Dade County. So then I spent 2 weeks in the Galapagos Islands with Dean Evan Del Sandro. I can tell you right now, if you're not registered to go to this school, register just to spend 2 weeks in the Galapagos. It is, I mean, with Professor Evan. It is amazing. And that's part of why it's amazing. Every morning, seriously, for 14 days, before we met for breakfast at 8 a.m., there's a lagoon, and you can swim with sea lions. And they swim with you, and they will just do their thing. Well, okay, so I come here. In order to graduate, I had to do an internship report. I had an incredible professor, Kelly Cox. She was general counsel at Miami Waterkeeper. She taught me environmental law. I asked her, hey, would you do me a favor and get me an internship? She said, no problem, come in. I came in the first day. I said, what am I going to do? They said, you're going to do a deep dive into septic tanks. I said, great. And then I left, and I went back home, and I said, I don't know what a septic tank is. And I grew up in Miami Beach, and that's a problem because Miami Beach doesn't have septic tanks. They used to. In fact, they used to in 1949, okay? Because this is what I found when I started doing my research. This is from Look magazine in 1949. If you want to have the original edition, I bought it on eBay. And look at the headline there, right? It says, the land of perennial summer is every man's ideas of a Garden of Eden, but thoughtless dumping of sewage and waste makes it one of our filthiest states. Well, that was 75 years ago. Guess what? It's the same today. And that's what I found out in my internship. So I studied septic tank. Back when I did it, I completed it in 2020. I didn't know about septic tanks, but most people didn't know either. And the government, who knew about it, knew very well about it, because I learned that in 1970, they signed an agreement with the United States government to completely eliminate 120,000 septic tanks. And the US government gave them $500 million, which in today's dollars is a lot more than the $4 billion it would cost to get rid of them all today. They took the money and they built 3 beautiful new central sewer plants that we still have today, unfortunately, those same sewer plants. And then they skipped one part. They didn't take out the septic tanks. They leapfrogged, and they created new lines so they could develop further west. And that's why all of the septic tanks that we have today are all along the beautiful coastline of the Biscayne Bay, and they're all along the canals, and they're in about an average sea level, groundwater level, about 3 to 6 feet below the ground. So they're completely dysfunctional, which is what I wrote about in my report. And then I handed it to my good friend, Alex Harris, who did a story in the Miami Herald, an expose, front page, talking about it. And lo and behold, the government didn't like that, because we called them out. And I don't know if you've heard, but now it's kind of the first topic of conversation, because they did not honor the agreement they made with the United States government, and we're paying the price today. The other headline that I just want to mention before I finish is we did when I was so then a couple of years later, after being an intern, they made me, they asked me if I wanted to be on the board of directors. So now I've been on the board since 2022, and I'm very proud of doing that. But one of the great things that we did that's a very current conversation is we spent a year doing a report card on the stormwater systems that serve Miami-Dade County, and guess what the average grade was for 34 municipalities? It was definitely below a C, okay? They would not have passed, and they would not have graduated Rosenstiel. Well, I just wanted to show this. So this is from Give Miami Day. These are the junior ambassadors that we run a program with. And that leads me to the introduction to Professor Macdonald, because I don't know if she remembers, but I got extremely depressed when I was in school, because I didn't know anything that I was learning about. I wish I didn't learn all about it. And I came to Professor Macdonald after class, and I said, you know, I can't believe what we've done to this, and it doesn't seem like there's an easy solution, and how are we going to get out of this? I'll never forget. It was after class, but you looked at the empty classroom and you waved your arm and you said, they're the ones that are going to do it. They're the ones that are for our future. So I thank you very much, and I'll give it over to Professor Macdonald.
Thank you. Thanks a lot, Aaron, and very glad that you were able to learn something at the school and use it now to help us protect this area. It's now my distinguished pleasure to introduce Dr. Catherine Macdonald. Typically when we recruit professors at a school like ours, we open the position internationally, and we are genuinely looking for the very, very best candidates. And occasionally, in fact, it appears that the very best candidates are in our own school. This is the case with Catherine. Catherine studied here in the shark program, or work on shark ecology and biology, and got her Ph.D. in 2017, right? Yeah, 2017, from the Abbess Center, as well as the Department of Environmental Science and Policy. And after working a little bit outside of the school, was recruited by the school as a research assistant professor to lead the shark program that we have. The shark program focuses on the ecology and the biology of sharks, and it offers multiple activities, both from the education point of view as well as from the research point of view. Catherine is also the track coordinator and leader for our marine conservation program. And in addition to that, if it was not enough, she's teaching multiple classes that are related, again, mostly to the ecology of the area. And the conservation, okay? So really she works at the intersection between the science and society and help educate our students in that field. In addition to that, Catherine also created, or she's the co-founder of the field station, which teaches students marine science fieldwork skills in a safe, supportive, and inclusive environment. And that offers many of the younger professionals an opportunity to study and to learn field methods that are very, very valuable for their future career. So without further delay, I forgot to mention, by the way, that I spoke a little bit about the education, but Catherine is also a brilliant scientist. She has already published about 40 peer-reviewed publications, which is exemplary, I would say, for a junior faculty. So Catherine, thanks, and the floor is all yours. Thanks.
Hi, everyone. Thank you so much for being here. I was imagining that this would feel like teaching a class, which I do in here 4 times a year. But those classes are 40 students, so it feels a little bit different today. I am so grateful to have you all here and to have a chance to talk to you about sharks here in South Florida, in part because I think that this region is incredibly important for sharks, but also is truly at the ground zero for changing climate. So a lot of the work that we do today focuses on sharks and how their habitat use, physiology, and lives are going to change under conditions that we're already starting to see even in my career. I'm especially grateful to all of my students, past and present, who are here today, including Erin, because it's very nice to have those friendly faces in the audience. So a little bit about who I am, although I feel like you already have a sense of it. I did my Ph.D. here at the Abbess Center. I started teaching at the University of Miami's Rosenstiel School because somebody got another job at the last minute and they needed a last minute cover. And Erin was actually in that very first class that I taught. And I'm going to hold you responsible here. He and Wayne and a few other of those early students really convinced me that teaching at the Rosenstiel School was what I wanted to do. So thank you all for that. I started as a part-time lecturer, teaching just a couple classes a year. I became full-time in 2020 and became a research assistant professor here in 2022 and took over leadership of the Shark Research and Conservation Program in 2023. So we're staying very busy. In 2021, I became a National Geographic Explorer associated with my work on hammerhead nurseries here in Biscayne Bay, which we'll talk at least a little bit more about later, although I started out with at least an hour and a half worth of slides, and they told me I have 35 minutes, so I made the cuts before. They're not in there now. All right, so when I tell people that I am a shark scientist, they usually imagine something pretty much like this. And when I talk to them about what I actually do, it looks a lot more like this. Our lab works on mesopredatory and juvenile sharks, in part because they get so much less research and conservation attention than their larger relatives. Everybody wants to talk about the big sharks. Discovery Channel wants to make their shows about the big sharks. But it turns out that if you don't have little sharks, eventually you're not going to have big sharks either. So we especially focus on these smaller bodied animals and their specific conservation needs. And one of the reasons that I ended up with that focus was that Biscayne Bay, as I started working here, increasingly it became so clear that it's very important to these species, to these juvenile life stages. The other thing that people think when you tell them that you are a professional marine conservation scientist is that all your work is in places that look like this. Beautiful, untouched by human hands. You're the only person to ever have snorkeled this incredible, vibrant coral reef. And the main places that I work look more like this. So a little bit this talk is more about how to appreciate what we have right here in our backyard than it is about how amazing sharks are somewhere else, where the water is a little bit cleaner or has a little bit less fecal coliform bacteria. The Shark Research and Conservation Program is now, as of 2023, a part of the Robert K. Johnson Center for Marine Conservation, which is a new center here at the Rosenstiel School that we're very grateful to be a part of, in part because it is distinctly focused on fostering connections between science and society that inspire and transform marine conservation in Florida and around the world. And so it was a really natural fit for us because we fundamentally are concerned with applied conservation research, understanding the biology and ecology of sharks with the specific intent of better protecting them. Education, we take about 800 students a year out on field trips at middle and high school ages to get hands-on with sharks and STEM. And our outreach and engagement work, which involves teaching younger students who were not quite ready to put next to a shark yet. So in 2024, a little bit about what we've gotten up to. SRC was home to 28 graduate students in 2024, the cohort that graduated and the folks who are still working. We tagged 566 sharks last year across 115 days of fishing. We brought 788, but I rounded it up, middle and high school students shark tagging. We ultrasounded 54 of those sharks to check out whether they were pregnant or not. If you're wondering why did we ultrasound so many fewer than we caught, a big piece of that is if it's not a mature female, we probably don't need to ultrasound it. We deployed many acoustic tags, and we have detected 500, 2,022 pings from those tags. We flew 251 drone flights, a lot of which were Noah Sonnenberg right over there, who is wrapping up his MS, which I'm also going to talk about a little bit. We processed 404 stable isotope samples to study diet and feeding ecology, 200 hormone samples to study reproduction. That's Ph.D. student John Flavin and master's student Katie Miller. And 1,700 parasite slides stained and fixed in the field. So drop of shark blood onto that slide, smush it one cell layer flat, stain it and save it to look for tiny parasites later. We're going to talk about that one too. And that's MPS student Haley Aker and MS student Charisma Acosta. So obviously, we have a big team that's doing a lot of different things, and I'm going to talk about as many of them as I can cram in to 30 minutes. We also have the FINS program, which is focused on bringing girls into STEM. So only 25% of the STEM workforce today is women, and in many cases, girls lack female role models. I can tell you that when I started my career, I had never heard of a single female shark scientist who was senior to me in the field. And even now, less than 10 years out from my PhD, I'm probably the 5th or 6th most senior woman shark scientist in the United States. So our field is very male-dominated. And so through this program, we bring girls into the field and the lab with women scientists and graduate students. And we're launching a new semester-long program this semester with support from NOAA and the National Marine Sanctuaries Foundation to give them a semester-long experience coming into the lab, going into the field with us, and having the chance to really connect with grad student mentors. I would also like to thank the Wiggin family for their support of that program. So let's get a little oriented. We all know where we're talking about, Biscayne Bay, Florida. We could throw a rock and hit the bay from here, pretty much. But what we might not know is that South Florida's estuaries are warming 6.9 times faster than the global ocean. That means that my shark nurseries are going to tell us about what happens when shark nurseries start heating up before almost anybody else's will. Which is both a tremendous privilege and also sometimes something that's quite difficult to have to supervise as part of your career, right? Watching habitats you care about decline, watching species that you used to see in good health start to fail can be really difficult. The great news that I have, other than that there are a lot of amazing students here working to solve those problems, is that we can do better. It wouldn't even be that hard. And so we're going to talk a little bit about the challenges they face, and then maybe in the Q&A, if somebody wants to ask me about what we can do better, Aaron and I are happy to talk about that too. The waters around Miami likely serve as mating, gestation, so you're pregnant, you're working on them, pupping, or nursery habitats for at least 10 species of shark and ray. Why do I care about juvenile sharks? Why do I care about reproduction? A lot of shark research focuses on those big apex predators, not those mesopredators, who are 1 or 2 steps down. But juvenile and small sharks are fundamentally different from adults. If you are developing a plan to conserve tiger sharks that thinks about conserving adult tiger sharks that make massive long-range migrations, you are not thinking appropriately about making sure that there will be massive tiger sharks in the future, because it turns out that many of those subadults depend on these shallow coastal habitats that are under threat. Biscayne Bay is also the first described nursery for great hammerhead sharks on the east coast of the United States. And that paper is one my team published in 2021. We've hit you with the QR code if you want to go ahead and read it, although I don't know how many of you are eager to go read a scientific publication. So I'm going to talk in a little bit of depth about one project that I'm really excited to be sharing because it just got published last week, looking at juvenile nurse sharks and their capture stress. And this is the work of 2 master's students, Catherine Giese, whose first author publication just came out, and I'm going to make y'all clap for her. Thank you. And Chris Will, who's still working on his manuscript, so watch out, Chris. I've got this whole audience on my side. So nurse sharks are a great species to study if you're trying to learn more about stress, because nurse sharks are impossible to kill. In my entire career, I have heard of one nurse shark dying as a result of being caught, and it swallowed the hook and then managed to stab itself through the heart with it. Nurse sharks are incredibly tough, so they're a great model for stress because you're not going to harm them by accident while you're trying to learn about their stress response. They're a very common coastal benthic species here, and they're pretty sedentary. They don't move around a lot. They're kind of the couch potato of the shark world with low metabolic rates. They interact quite often with recreational fishers. One of the things that my lab is working on is better understanding their habitat use as juveniles, and pretty much it seems like they rely on quite small areas, and so where their core habitat overlaps with recreational fishing hotspots, they'll get captured more than once. My 2 master's students, I think the most that they caught a single nurse shark was 5 times in a month. 5 times a month is a lot. So we started to worry, what effect are these repeated captures having on their well-being, on their growth, on their long-term health? In general, scientists think that nurse sharks are quite resilient to capture stress. That's a bit of an understatement. And they're really pretty abundant, but there aren't a lot of studies on them. And so we felt like they were the perfect place to focus this study. Here's Catherine. Working on her project. You can see that we were collecting this data during COVID. And for her project, we caught 27 juvenile nurse sharks, varying in size from 63 to 153 centimeters. So like pretty small guys, not very big, in water between 70 and 93 degrees. So as soon as one was caught, we would draw a small blood sample. Then we'd put it back in the water, still on the hook, and make it fight us for 30 minutes. And we would check in on it every 5, 10, or 15 minutes in variable orders. So shark 1 might get caught, get let go, 15 minutes, 5 minutes, 10 minutes. Shark 2, 5 minutes, 15 minutes, 10 minutes. We're mixing up the kind of amount of time between sampling so that we're controlling, we're not putting that error in our data in terms of how often they're being handled. If you're thinking, this sounds mean, we felt kind of mean. They kept trying to just lie down on the bottom, and we'd be like, no, you have to swim. But what we found was more or less what we expect, which is something that scientists love. Their lactate, which is just a simple measure of how hard they've been working, right? The stuff that makes your muscles burn when you work out. It's formed by the breakdown of muscle glycogen, right? But you don't have to know that, right? They're feeling the burn. The level of lactate in their blood is predicted by how long they've been fighting. Right across that 30 minutes, we see it go up. By water temperature, which makes sense to us intuitively, right? Because water temperature is tied to metabolism in sharks. Unlike us, they don't regulate their body temperature. So if you heat up the water around them, you heat them up. They burn a little bit hotter. And by species, which this we know from the literature, nurse sharks are considered a pretty low response species. We also know from the scientific literature that lactate levels predict post-release behavior and mortality. The higher an animal's lactate level, the worse it is at swimming away at the end of that interaction with a fisherman, and the more likely it is to die after capture. Then we did this analysis by temperature, and you'll see that the higher lines are mostly orange and red, and the lower lines are mostly yellow. So the hotter the water is, the more pronounced that stress response that we're seeing from those individuals is. This study was great. It was tidy. It showed us exactly what we expected. So of course I had to go and ruin it by having another student take a next step. So the next step we took was trying to recover these sharks, right? So we're catching it, we're cutting the hook, and we're immediately letting it go into a recovery pen. And what we want to know is how long does it take you to get over having been hooked? And this is literally a best case scenario for being caught, right? Because the entire event is less than 90 seconds. They're cutting the hook instead of ripping it out of the animal's mouth, which a lot of anglers do. So 33 sharks underwent this treatment. I think that the thing I can say about both of the master's students on this project is they have some real fortitude because especially in the winter months, you can fish a lot for nurse sharks without catching a lot of nurse shark. But they caught, for this project, in 71 to 93 degree Fahrenheit water, put them in their pen for between 30 minutes and 8 hours in 30-minute increments. So basically, we dropped you in this pen. We're going to come back to you in 2 or 3 hours, pull you up, see what your lactate levels look like. And now science has betrayed me and not given me the result I expect. We see peak lactates about 4 hours later for all 3 temperature groups. But we see the highest lactate in the lowest temperature water, which is the opposite of what we expected. I have 2 possible theories. One of them is in that hotter water, that juiced metabolism lets them process the byproducts of that fight and that stress faster. So you never see it reach the same level in the blood as it would at a slower processing rate. And my other possibility is that maybe we're experiencing more food stress in the winter, and so we're under nutritional stress already, and we feel less well-equipped to handle capture. So that's a next and future problem for my lab. But science doesn't always give you the answers that you would like, but it does usually give you answers that raise more questions. Right now, we're working on redeploying adult nurse sharks with accelerometers on their line to try to replicate this recovery experiment with like angry 9-foot nurse sharks. When I proposed this project, one of my captains was like, do we have to? And I was like, yeah, we're going to do this. It's going to be great. I'll get back to you all on that one. We're also really interested in habitat and chronic stress effects. So as your habitat quality gets worse, do you muster less of a stress response because you're hungry and in worse condition? Or do you muster a stronger stress response because it's additive with the other stressors you're experiencing? I'm interested in how much trouble they're getting themselves in if they're really attached to a recreational fishing site. How many times might you get caught? And we're very interested in changing water temperatures, including climate change. We have some big grants we're applying for that I'm hoping will let us study this in a lot more depth. I'm also really interested in understanding whether age and life stage affects your stress response in other species, which is something we don't know much about in sharks. Do juveniles find it more stressful to be captured, less stressful than adults? Almost all of the regulations that we have are, at best, species-specific. They're not concerned with how old you are. But of course, we know there's big differences between babies and adult humans. And we can probably expect that the same is true for sharks. All right, I'm going to let you watch this video while I sip some water. So this is directly off our research boat, She pretty much ran our photographer over when we let her go. But we will touch a little bit on our great hammerheads and a few other species. So I'm going to grab bag you quickly through a few different projects. So stay with me. I appreciate you. So we have a lab project in collaboration with the Marine Megafauna Foundation studying the Atlantic guitarfish. It's the only guitarfish species we have here in South Florida, and it's one of the most threatened of the sharks and rays. Guitarfish are technically a ray, but we're close relatives. So I have an amazing master's student, Kylie Powers, who defended last semester, who has used their distinct spot patterns on their rostrums to individually identify them and study their movement in South Florida based on re-sightings at different locations. So that's the animation that we're watching over here. And what we're going to see is that a lot of guitarfish are not huge travelers, but that every now and then they're covering distances that we're a little bit impressed by. The purple one that looks like it's crawling across land, that's just an artifact of the fastest straight line from one point to the other. I promise it stayed in the ocean. So what her data is telling us is that overall, they're pretty site attached. For most of them, where they are resighted, we're seeing them fairly close to where they were before, which makes sense because they're about this big. And they're like flat, weird little shovel pancakes. But we have detected a few surprisingly long movements, including detecting one of the acoustic tagged ones on a receiver of our own down here in Miami. So we had an animal make a round trip from West Palm to Miami and back in about 15 days, which for a ray this size is really a pretty impressive swim. We know next to nothing about them. Literally, if you look them up on the IUCN Red List, which is kind of the best authority for this, they say that they don't occur here. That's how little we know. But we have them making these long-range movements as well. So Kylie is using photo ID, and Jenny is using acoustic tagging to try to better understand their habitat use, their site fidelity, and the distances that they might travel. And one of the things we hope to do through this is better understand small guitarfishes in a place where they're not fished, so that some of that data can be extrapolated to places where they are, because they're commonly fished for meat in other parts of their range. We also have projects on their diet and stress response. That's led by an undergrad in our lab, Ryan McMullen, in collaboration with Ph.D. student John Plavin. And then I'm going to skip off the quite cute Atlantic guitarfish and talk briefly about parasites, because we talked about them before. We know parasite loads on sharks and fish can increase as a result of warming and water pollution. Bad water quality isn't great for sharks, but it can be good for creepy crawlies. So we study parasite load over time. That's a couple different students working on that project. We are looking at ectoparasites, the ones that are on the outside, endoparasites, the ones that are inside, how the ones that are on the inside got on the inside in the first place. That's vectors. Charisma acosta is studying trypanosomes, which are like a little corkscrew worm-looking thing that we find in those smears, right? One cell layer of blood, little snake. And we think that they are transmitted by leeches, but we haven't yet been able to catch any leech that's carrying them. So it's still kind of an open question how they move from shark to shark and complete their life cycle. We have a project looking at changes to shark skin microbiomes. We swabbed. You see this like weird, gross, fraying, lumpy looking edge to this fin here? I tried this on someone else and they were like, I don't know what a normal shark fin edge looks like. If you don't, it doesn't look like that. That's like gooey and frayed. It doesn't look right. So we swabbed these lesions, which are created by a flatworm that we're looking at up close up there. And we looked at how they might be different in terms of their microbiome than healthy areas of skin. And what we found was a nasty named tenacibaculum, which has only ever been seen in sharks in captive species, is leading to this skin degradation and fraying in our juvenile sharks. Over the last 5 years, we went from essentially never seeing these skin lesions and this damage to fins to seeing the lesions formed on about 60% of our blacktip juveniles, including animals that were less than 2 weeks old. So these changes in parasite load might not be a big deal for a 6-foot shark, but for a 50-centimeter shark who's a newborn, it's not really clear to us how harmful these changes in parasite prevalence are going to be. Noah, who I shouted out before, is using drones and hormone analysis to study bonnethead shark aggregations. And he's seen some crazy things, including aggregations of a size that shocked me. What is the most you've had in a day?
It was 117.
117 individual bonnetheads swimming together in one day. I didn't know they did this. The published literature doesn't know that they did this. It says, oh yeah, they form groups of like 10 to 15. No. Here in Biscayne Bay, we have them forming these enormous social groupings that are influenced by lunar cycles, and he's observed multiple mating attempts, which is what we're watching here on this video. We see that flip. So sharks are not very romantic, but when a male shark really likes a female shark, he tries to bite onto her at her pectoral fin or on her back, flip her over, and use his clasper, which is a modified calcified pelvic fin, to deliver sperm that will allow her to make more bonnetheads. So we see this exaggerated following behavior. We see like S curving after. I know that it's a little hard to see, but we can, right?
Yeah.
And then we're going to watch the boy at the back try to bite on and flip her over, and she is not having it. But that is exactly what shark mating looks like, and to the best of our knowledge, this is one of the first ever observations of wild bonnethead mating. So while we don't, as far as we know, have baby bonnetheads commonly here in Biscayne Bay, or at least we don't catch them on our gear, we know that they are mating and gestating in this habitat, which is likely important to bonnethead populations. Finally, I'm going to talk a little bit about this research, which is my Ph.D. student John Flavin's work. This is going to get a little technical, and we're going to try not to go too far into the weeds. So he does something called multi-tissue stable isotope analysis. And what you need to know about that to stay with us is that he's using 2 different tissues, plasma and muscle, and that they have different incorporation rates. So if you change your diet, it's reflected in your plasma way before it's reflected in your muscle. You replace your plasma more often. Stable isotope analysis is just a tool that lets us learn about where an animal is eating. Is it an apex predator, a mesopredator? How high up on the food chain? And what habitats the photosynthesizers that support the food web it's eating in came from. And that sounds complicated, but really it just means one signature tells us you're eating things that come from the bay, and the other tells us you're eating things that come from offshore pelagic environments. We with me so far? Up and down trophic levels, inshore, offshore habitats. So this can help us answer questions about what an animal's eating and where it comes from. We caught 50 great hammerheads across sizes from what we call young of the year animals, animals that were born that season, through big adults. And we got a white muscle biopsy and a plasma sample. For 37 of those individuals, we have both. You're going to be not that surprised to learn that we have more of those samples for smaller animals, because as you get bigger, you're more able to tell me no when I ask you for a muscle or a blood sample. What we're looking at here in this figure is data that's telling us that the smallest individuals, which are in green on the right, have inshore signatures, right? We're living mostly in the bay. We're relying on bay food webs, probably. But they're not very different in their trophic position from the other 2 circles, right? So we all are pretty high-level predators. The second group in blue, which is the kind of like fatter circle, oval, is our medium-sized individuals. And that's telling us, hey, as we get bigger, we really expand our diet, right? That sort of moves up, but it kind of moves down too, and also out, and we're moving into offshore habitats. But our overall, what we call a dietary niche, is just bigger. we probably eat a lot more different things. And then our biggest individuals, the long circle, again, really big, probably a pretty broad diet, but we see that at least some individuals are at a slightly higher trophic level overall, and that we're still using those bay habitats as a source of food, in addition to using offshore and reef-associated habitats. So John used these paired samples to do a really cool analysis, and what he found is that we see a habitat shift in their resource reliance between the dry season and the wet season. So we see them moving, right? In the dry season, we're hanging out in the bay. We're happy there. It's all good. And then we make a move offshore, and then we come back. And we notice that this is, do we see the blue ticks at the bottom? We can see them? That's our catch of adult great hammerheads. And so we see that our expansion out of the bay is associated with times when there's not a lot of adults around. These juveniles know that an adult great hammerhead thinks a juvenile great hammerhead is a decent snack. And so one of the shapers of their habitat selection looks like it's the presence of adults, with avoidance of direct overlap or direct competition with those adults as a potential driver of their habitat use. So John came back with this analysis, and we were really excited about it, but we were also, I don't want to say not sure, but you're a scientist, you always want to triangulate on a hypothesis. And so we went to our acoustic tag data. This is our Nat Geo funded research, right? We're putting these 10-year acoustic tags in juvenile sharks, great hammerheads, and learning about how their spatial habitat use changes as they age. So we went back to that data, and you're looking here at a small, medium, and large-ish juvenile, right? So the blues are big juvenile, and we see we're all in those coastal habitats. But our smaller girls are sticking around the nursery right here off Key Biscayne. And then boom, the bigger one makes a few migratory moves to our receiver offshore. And it turns out that they coincide perfectly with the timing of the dietary shift that John detected. We love it when science works. This is just another way of representing that data, where we can see that our biggest individual, bay habitats are warm colors, offshore habitats are cool. We see our detections of our biggest individual are almost all offshore somewhere. And our juveniles that are a little bit smaller mostly depend on inshore habitats, with just occasional pop-ups on the ocean side of Key Biscayne. I have a lot of people that I want to thank, but I'm going to leave you with this picture of my students and field team. I'm very grateful every day to get to work with all of the students here at the Rosenstiel School, and especially the amazing team that makes up my lab. I would also really like to thank the faculty at the Johnson Center and in my department, the Rosenstiel School as a whole, which has made me feel like a welcome part of their family for the last 15 years almost. Our scientific collaborators and funders, some of whom we have acknowledged here, and especially all of you for coming out on a Monday night. Thank you all so much for being here.
Thank you, Catherine. I wanted to say your work is so interesting, and look at this great audience. Thank you, audience, for coming out tonight. We wanted to tell you, it's great that you're here engaging our students today and the young students that you work with on all the trips that you take out. I mean, you're an inspiration. I just wanted to say, before we get on to the question and answer portion, we're going to have people, we're going to walk around with our microphones. You'll raise your hand. But before we do that, I have to make my pitch. So you saw we have great alumni here. If you'd like to support our students at the school, you can reach out to me. My name is Jennifer Dillon, and my email is supposed to be up here. It's jsdillon@miami.edu. If you'd like to learn more about the Rosenstiel School and the work that we do, you can find us online at earth.miami.edu. Our students, depend on resources. We are a private university, so if you would like to support our graduate student fund, our undergraduate fund, or our opportunity scholarship, you can speak to me and I can help you make that happen. So thank you, and I think we'll go ahead and start with questions. Anybody want to take this?
Throw up a hand if you have a question.
Just raise your hand and we'll bring the microphone around.
Thank you so much. This was fascinating. I think you should become a TED talker. I really enjoyed the talk. I'm wondering, in terms of the stressors that you look at, did you do sort of temperature is an obvious one. Did you look at other ones? Ocean acidification comes to mind, or changing of currents, or intensification of storms and things like that. And also, how does that compare to like maybe a baseline evolutionary development?
That is a great question. Everybody could hear? So one of the really hard things about trying to study global change and sharks is that there's so many global changes and they intersect with each other all the time. So there's a bunch of captive studies that look specifically at acidification or warming, mostly for small egg-laying species from the Indo-Pacific, because they're a lot easier to work with in captivity than some of the species that we catch here. I think about the contribution we're uniquely positioned to make as being able to ground truth some of that. Because somebody tells me, oh, their body condition is a little bit worse when you make it hotter, right? It affects their metabolism. Yes, I don't doubt it. But that's one of 20 things that we're doing that makes Biscayne Bay a more challenging place for them to live. And so we come at these questions often from the perspective of outputs, right? Where I can best answer a question about how well a nursery is functioning by looking at the health and survival of the juveniles that are in it, rather than trying to tease apart every individual change that's occurring in the nursery, just because from a scientific perspective, that's probably beyond our capabilities at this point. So a little bit, I think of our small sharks as warning indicators for some of the environmental changes that we're making, because I do, on the worst days of my job, I worry that I'm going to be the scientist who is the first to measure the loss of a nursery to climate change, because our juveniles are struggling more now than when I started collecting this data in 2016. But I also do think they are incredibly resilient and that we have the capacity to turn it around.
Do you find the movement of sharks in your study attracted to or repulsed by polluted areas?
I think it varies not only across species, although definitely across species, but also even across individuals. So we know that sharks learn, right? We know baby sharks get better at hunting as they age because they get more practice and they watch what other sharks are doing and they copy them. And so there's some tiger sharks that I would say almost specialize in being like, that gross canal, that's my gross canal, and I go eat the gross stuff that comes out of it. And then there's other sharks that really don't have that level of tolerance for more polluted waters, either because they have higher oxygen needs, and so that pollution can interfere with their ability to breathe well, or because they don't prefer to eat that stuff. I can tell you, I had a tiger shark in the last year vomit up a cormorant on me, a pig's foot that I'm pretty sure a fisherman gave to it. Like, I don't think it went out and hunted a pig, but it was a trotter, like rotting sea turtle, a lot of feathers, like a surprising number of feathers. So some species, whatever's out there, they're excited. And others are really a lot more selective, both about the habitats they'll tolerate and the foods that they're looking for.
Hello, good night. I have a question. You were talking about the parasites. How do they manage these parasites to live longer, these sharks, especially the small ones?
how do they stay on there? Or?
Yes, how do they manage these parasites to I know people would take medications, but what do you do to manage these parasites?
Yeah, I mean, in some cases, I think that an animal is big enough and inhospitable enough to the parasites that it sort of takes care of itself. So a lot of species do produce a slime coat on top of their denticles, their scales. And so they can kind of make extra slime and try to slide the parasites off. They have immune defenses in some cases. We have drone footage of sharks rubbing their bodies on the sand to try to scrub those parasites off. So they have strategies to manage them, but I think that those strategies probably vary in how successful they are. We catch a lot of animals that have 1 or 2 parasites where you don't feel like it's a big burden on them. But we do see maybe between 5 and 10% of animals every year that are carrying enough parasites that you start to think that looks like it hurts, that looks like it would be hard. And for those, I'm not sure that their traditional mechanisms for managing those parasites are still working that well in modern oceans, because we also know that warming can juice parasite metabolisms so that they can actually reproduce faster, increasing the risk to animals.
Hi, I'm Sarah. Thank you for this evening. Quick question. How would a high school biology teacher get in contact with you to propose maybe students to come and tag sharks?
Absolutely. If you shoot me an email, which is on that slide, but it's just my name.
I already photographed it.
Cool. Send me an email and I'll connect you with our logistics coordinator.
Hi, what can I do about the rising temperatures? How can we make it better?
So I think about this in part strategically from the perspective of what other problems can I solve for them, knowing that I individually am not going to reduce climate warming, right? So if we can improve water quality, as Aaron told us, that's a very doable thing to do, right? Those improvements in water quality can take a lot of stress off them. We can restore and protect existing habitats better than we are, right? Miami does not necessarily do the best job either of enforcing boating regulations in ways that protect the integrity of seagrass meadows, fining property owners when they cut down mangroves, which are really important to a bunch of different juvenile shark species. So climate can feel a little bit hopeless, but I actually think that the warming that we're talking about wouldn't be an unmanageable problem for most of our juvenile sharks if we were able to reduce some of the other stressors. So I approach it from how can we make Miami's ecosystems healthier, more resilient, better places to live, and how can we do our best to ensure that they stay that way, because there are a lot of proposed kind of coastal engineering solutions to protect the city from sea level rise that involve building concrete barriers, in some cases right through nurseries that we've described. So trying to find ways to build green infrastructure that can support the species that are here, in addition to helping manage the risks of climate change for the city, is about the best answer I have. But the really shortest answer I have is people will sometimes ask me at these talks, why should I care about sharks? And you all came out to listen to me talk on a Monday night, so Tuesday night, so you care about sharks. But I'm not telling you you should care about sharks. If you don't care about sharks, you don't care about sharks. That's okay. The thing that I want people to do is care about something, right? Care about septic tanks. Care about dolphins. Care about whatever you care about. But I think that the problem isn't that everybody's not focused on conserving my shark nurseries. I think the problem is that everybody's not focused on figuring out what's important to them and acting to save it. And I think if everyone was doing that, my job would be a lot easier.
I think that we're over time here, but I wanted to say thank you, Catherine, so much for your time tonight. Thank you, Aaron, too.
Yes.
Before everybody leaves, I want to mention we are going to have this available on our YouTube. It'll be on Facebook as well, probably next week or so. Please take her information here. If you have extra questions and you didn't get to ask it tonight, you can send her an email. And also our next speaker is Esther Guzman, Dr. Esther Guzman. She is coming on February 6th, and we hope to see you all, and have a good night. Thank you.
Thank you for being here.